Okay, all right.
So just so you know, Dr. Foster, we have not started yet, but the stream is live, and folks should be able to hear you now. And I know we're waiting for the ranking member.
This hearing will now come to order. Without objection, the chair is authorized to declare recess at any time. Before I deliver my opening remarks, I wanted to note that today the committee is meeting both in person and virtually, and I want to announce a couple of reminders to the members on the conduct of the hearing. First, members and staff who are attending in person may choose to be masked, but it is not a requirement. However, any individuals with symptoms, a positive test, or exposure to someone with COVID-19 should wear a mask while present. Members who are attending virtually should keep their video feed on as long as they are present in the hearing. Members are responsible for their own microphones. Please also keep your microphones muted unless you are speaking. Finally, if members have documents that they wish to submit for the record, please email them to the committee clerk, whose email addresses was circulated prior to the hearing. Well, good morning and welcome to our members and our panelists. One of the most important functions of the Science Committee is to help inform Congress of rapidly emerging technologies that will have important policy implications, so that we're not always playing catch-up. The past decade has seen progress in medical technology that would previously have been seen as science fiction. Not long after its discovery in 2012, I started getting increasingly urgent requests for meetings from cellular biologists telling me about the discovery of an incredible gene editing technique called CRISPR, and that brave new world was not going to be a century away and might not even be a decade. Many of us were here for the 2015 Science Committee hearing on the science and ethics of genetically engineered human DNA. In fact, it was only 3 years after that, 6 years after the discovery of CRISPR, that a Chinese scientist shocked the world when he announced that he had created the first gene-edited human child. We are still grappling with the potential societal, ethical, and economic implications of that breakthrough. I believe that the aging therapies being evaluated by the geroscience community today may be equally seismic in their impact. The hypothesis at the heart of geroscience is that aging itself is a relatively small set of general processes and that some of them may be malleable. It is a hypothesis that is reinforced by the wide range of aging processes observed in the natural kingdom. We didn't used to think of aging as a disease, but that may be changing. Rather than looking at individual conditions, the entire process of aging is being considered as a driving factor behind increasing morbidities. And because of the analytical tools that have been developed through decades of federally funded research, scientists now have the ability to break down aging into a collection of biological events and developing a deep knowledge about how it happens at the cellular level. They are making connections between these cellular changes and how they manifest as illnesses and pain throughout our aging bodies. They're learning that these biological events at their most basic level may be influenced by deliberate or even inadvertent intervention. And if you can do that safely, aging and the goal of increasing healthy lifespan or health span may come within sight. In just the past 3 years or so, scientists have started testing aging interventions on humans through small FDA-approved clinical trials. The first trials had fewer than 100 human subjects, sometimes fewer than 10. But right now, the Albert Einstein College of Medicine, with the support from the National Institute of Health, is standing up a clinical trial with a cohort of 3,000 subjects to evaluate whether a prescription drug called metformin can actually help delay age-related chronic diseases in general. A formal trial like this that seeks to solve for several otherwise unrelated diseases is relatively unprecedented. Imagine how profound it would be if we could identify a drug or therapy that can simultaneously mitigate Alzheimer's, cancers, macular degeneration, hearing loss, and joint pain with a single or a small set of treatments. Among other things, this would have an enormous implication on the federal budget. Of course, we're not there yet. This field is still nascent enough that the leading thinkers are still sorting out some of the basic vocabulary issues. It seems like almost all of the serious research efforts are focused on expanding the human health span, not the absolute limit or duration of human life. We will hear from our witnesses about the concept of health span today, but to be sure, the definition of healthy varies from person to person. If increasing health span is the goal, how will scientists know that they've done it? And how can the field determine the success or failure of an aging intervention without waiting decades to see how people fare as they age? If every experiment takes 70 years, this field will take a long time to develop. If researchers are ultimately successful in translating the outcomes that they have seen in model organisms into humans, we will have even bigger questions to confront. For example, if you extend health span, do you also extend the lifespan and simply delay the protracted aging process to a later date? What happens to healthcare costs and the burden of our healthcare system? Would we see people in their 60s starting second careers? And what would that mean for the broader labor force? Can insurance companies change your premiums based on whether or not you take the aging therapy? So we've all witnessed firsthand the breakneck speed of technology and technological innovation in the country over the last 40 years. And so we should know better than to be caught unaware. Our responsibility as policymakers is to get educated today on a field of research that could soon lead to transformational change. Our witnesses today represent the leading edge of geroscience, and I know that they will be faithful to the guidelines on our path to understand this topic and its implications at a deeper level. I now request unanimous consent to include in the record for this hearing a letter from the Buck Institute for Research on Aging, and without objection, so ordered. And the chair will now recognize the ranking member of the Subcommittee on Investigations and Oversight, Mr. Obernolte, for an opening statement.
Thank you, Mr. Chairman. I am so excited about this hearing. I'm excited for a number of different reasons. For thousands of years, mankind has been searching for the fountain of youth. And in the last several hundred years, it has become abundantly clear that the fountain of youth is going to be discovered not through exploration, but through science. It is a particularly relevant topic for this subcommittee to be taking up for several reasons. First of all, the fact that a lot of the research in geroscience is funded through the NIH and the NSF, which are governmental agencies that we have influence over. So it will be very interesting to me to see some of the fruits of that investment, and that will better prepare us to make the argument for continued investment and maybe greater investment in the future. But I am also interested because, as the chairman mentioned, the implications of a longer human lifespan are profound in the fields of public policy and governance, both good and bad. And it behooves us to start having that conversation early as we prepare for the societal changes that will come as a result of a greater life expectancy in the United States. But the third reason that I am so excited about this is because in many ways you can judge our progress as a society and a country by our average life expectancy. It's an indication not only of our prosperity, but also of the ways that we honor and treat our elderly. And if we can catalyze a future life expectancy and a future growth in life, in health span, the number of years that we have of productive, healthy living, we will progress not only as Americans, but also as a human race. So I'm very excited to hear from our witnesses and very excited that this hearing has been convened. Thank you, Mr. Chairman. I yield back.
Thank you. And if there are members who wish to submit additional opening statements, your statements will be added to the record at this point. And at this time, I'd like to introduce our witnesses. Our first witness is Dr. J. Olshansky. Dr. Olshansky is a professor in the School of Public Health at the University of Illinois at Chicago, research associate at the Center on Aging of the University of Chicago, and chief scientist at Lapidus Solutions Incorporated, a company he co-founded. Dr. Olshansky's work is focused on linking the scientific study of aging with investments in longevity and mortality-related products. Additionally, his research includes exploring the health and public policy implications associated with individual and population aging. Dr. Olshansky is also a board member of the American Federation of Aging Research. After Dr. Olshansky is Dr. Lauren Niedernhofer. I hope I did that right. Okay, thank you. Dr. Niedernhofer is a director of the Institute of the Biology and Aging and Metabolism and Medical Discovery team on the biology of aging. She is also a professor in the Department of Biochemistry, Molecular Biology, and Biophysics at the University of Minnesota. Her research program is centered on studying fundamental mechanisms of aging and developing therapeutics to target them. She's also contributed to the discovery of a new class of drugs called senolytics. Dr. Niedernhofer currently serves on the Advisory Council of the Division of Aging Biology at NIA and on the Board of Directors of the American Federation of Aging Research. Our final witness is Dr. Steve Horvath. Dr. Horvath is a principal investigator at Altos Labs and a tenured full professor in human genetics and biostatistics at the University of California, Los Angeles. His research lies at the intersection of several fields, including epigenetic biomarkers of aging, preclinical and clinical studies, genomics, and epidemiology. Dr. Horvath and his UCLA colleagues published the first epigenetic clock for saliva in 2011 and 2013, and he published the first pan-tissue clock, also known as the Horvath clock. Recently, he presented a universal clock that measures age in all mammalian species. Boom. Great. Okay, and now, as our witnesses should know, each of you will have 5 minutes for your spoken testimony. Your written testimony will be included in the record in its entirety. And when you've all completed your spoken testimony, we will begin with questions. Each member will have 5 minutes to question the panel. And if we have time, we may have a second round of questions this morning. And we'll start with Dr. Olshansky.
All right, first of all, I want to thank the committee for the opportunity to participate in these hearings on what I consider a new public health initiative known within the community of scientists and health professionals as geroscience. The story I'm about to tell you is an easy one to communicate because all of us are experiencing aging firsthand. In the modern era, most people in developed nations and a rising percentage of people in developing nations have the privilege of living a long life, a privilege denied to most throughout history. Pioneers in public health, medicine, and science from just a few generations ago gave us the gift of a long life, and since then, humanity has worked hard to maintain this privilege and extend it to others less fortunate. Life expectancy increased from 1 year every 1 or 2 centuries for the previous several thousand years to 3 years of life added per decade in the 20th century. The chances of surviving to ages 65, 85, and 100 have never been higher than they are now. There is reason to declare victory in the pursuit of extended survival, but plenty of work remains to ensure this privilege is made available to everyone. This longevity revolution came with a price. The modern rise of cardiovascular diseases, cancer, dementia, Alzheimer's, and non-fatal impairments are byproducts of success, not failure. We just had to live long enough to see them. While risk factors hasten the emergence and worsening of these diseases, the biological processes of aging march on in the background, uninfluenced by treatments for diseases. Aging has become the most important risk factor for the diseases and disorders that occur today. The quest for aging therapies discussed in this hearing is at the heart of a new public health paradigm that has been in the works for the last half century, but which has gained traction just within the last few years. Here's the story in brief. Changes in our cells and tissues occur with the passage of time. We call it aging, but there's nothing magical about this since we see the same process occurring in our pets and automobiles. It was suggested in the 50s, 60s, and 70s that aging should eventually become the target of medicine and science, but too little was known at the time about how aging happens. Medicine and public health did what it could in the interim to devise ways to detect and treat diseases one at a time, as if independent of each other. And this was a logical next step in dealing with the diseases that appear in aging bodies. But this approach came with limitations that can best be thought of as a game of whack-a-mole. Knock one disease down and another appears shortly thereafter. The longer we live, the shorter the distance between these diseases. The science behind the how question in aging has advanced rapidly, which now makes it possible to pursue the gold standard in public health, which is to slow down aging itself rather than just treat its consequences. Geroscience has come of age. It is the culmination of decades of research. It is not a theoretical construct. It has been demonstrated in the laboratory that rate of aging can be modified in other species, which means rate control is possible in humans. The first clinical trials of aging therapeutics, known as geroprotectors, are already underway, and the FDA is fully on board with this approach, which is to prevent disease by slowing aging. The health and economic benefits of geroscience will be substantial. A cure for cancer would be welcome, but that's just one disease of many that plague older bodies, and a cure for cancer would only add about 3 years to life expectancy. A geroprotector will simultaneously lower the risk of all fatal and disabling diseases of aging simultaneously, which means even a modest effect would yield amplified health benefits. The cost savings in healthcare alone would amount to over $38 trillion for each year of life generated with geroprotectors. The primary goal of geroscience is the extension of health span, not lifespan. So these advances will not generate a fountain of youth in the colloquial sense, but it will fundamentally change what it means to grow old. We will remain younger longer, retain our youthful vigor for an extended period of time, and compress everything we don't like about aging into a shorter duration of time at the end of life. There will be challenges that accompany the generation of a healthier and more robust older population, but the most precious commodity that we cherish most, our health, will be the gift of geroscience. It's difficult to imagine any scenario in the future where the generation of a larger, healthy, older population would not be pursued, even if challenges appear along the way. This is just an introduction to geroscience, and I'd be happy to take any questions you may have. And thank you once again for the privilege of participating in this hearing. My written testimony will address all of these issues in far greater detail. Thank you very much.
Thank you. And next is Dr. Niedernhofer.
Well,
Whoops, I think you're muted.
all right. Good morning, and thank you very much for this opportunity to participate. The geroscience refers to the fact that advanced chronologic age is the greatest risk factor for most diseases. Therefore, developing therapeutics that disrupt the biologic changes that universally occur with advanced chronologic age is not only logical, but potentially highly impactful to human health. A geroscience approach is anticipated to impact the health of the elderly to a greater extent than curing any single disease of old age, and this includes Alzheimer's disease or cancer. Importantly, geroscience aims to extend how long individuals are healthy, not how long they live, what we refer to as extending health span. So geroscience is based on 3 facts. First, we are in an unprecedented period of human history in which the number of elderly is doubling and surpassing the number of young people. This establishes the need for a new approach to prevent our healthcare system from becoming overwhelmed and healthcare costs skyrocketing. Second, the majority of people over the age of 65 have 2 or more chronic diseases. Hence, curing a single disease of old age will not dramatically improve the health of the elderly. Third, chronologic age contributes to the risk of most diseases to a much greater extent than other risk factors that we are currently treating. Thus, therapeutics targeting aging biology have the potential to be not only useful for many diverse diseases, but also to be highly effective at doing so compared to our current first-line treatments. So today, geroscience, where it stands is there is ample evidence that certain molecular and cellular events occur in most, if not all, people with advanced chronologic age. There's also ample evidence that some of these events can be therapeutically targeted in humans as well as in disease models. We have in hand FDA-approved drugs that target and stop or even reverse these molecular and cellular events of aging biology. We also have extensive data from animal models, what we call preclinical data, demonstrating that geroscience-guided therapies prevent, attenuate, or even reverse age-related diseases affecting most organ systems. This includes heart disease, Alzheimer's disease, and diabetes. There exists at least one drug, we've mentioned metformin, that appears to simultaneously stave off diabetes, heart disease, cancer, and cognitive impairment. There are also numerous other candidate geroscience drugs already approved by the FDA and in clinical use that should be tested for geroscience approaches. There's a lot of activity in this space, developing therapeutic interventions that target aging biology, primarily in academic settings at this point, but gaining traction in pharmaceutical industry. So what I perceive as key barriers to progress are adequate funding to pursue geroscience research in a timely fashion, lack of physician scientists and infrastructure needed to support clinical trials in geriatric patients, lack of public knowledge about geroscience, and a lack of biomarkers that report how well an individual is aging relative to just using their chronologic age as we do now. The federal government could facilitate geroscience research by providing federal funding dedicated to supporting geroscience research across many disciplines. Support for training physician scientists knowledgeable about clinical trials in geriatric patients, funding and support to create the infrastructure needed to advance geroscience research, including sharing of biospecimen and data, and facilitating the collection as well as the dissemination of information across diverse race, ethnic, and socioeconomic groups. So what would be the societal implications? Well, although this is really not my area of expertise, I can offer my opinion. Aging biology affects virtually every aspect of how an individual interacts with their world: communication, transportation, housing needs, healthcare needs. The elderly exit the workforce while requiring significantly more help. As a number of chronologically aged individuals continues to increase, we as a society will have to accommodate all of these changes. And geroscience, though, offers an alternative approach where we aim to keep those of advanced chronologic age healthy, independent, active, able to work if they choose, and able to contribute to the economy. Given the wealth of scientific evidence supporting geroscience, I feel it would be irresponsible not to try this alternative. I personally cared for 4 parents and grandparents over the last 2 decades, each of whom had multiple diseases of old age, and I can attest it's time-consuming, costly, heartbreaking, but also robbing younger individuals of productively contributing to society. Thank you very much.
Thank you. And as someone who shared the experience you just mentioned with his own parents, it's an important thing to sort of the aggregate quality of life of being a human. And finally, we have Dr. Horvath.
Yes, my name is Steve Horvath. I'm testifying in my personal capacity as a scientist. I'm very honored to speak to the members of this committee today. I would like to speak to you about a new class of molecular biomarkers known as epigenetic clocks, which allow us to measure aging in all mammalian cells, tissues, and organs. Epi means above, and it relates to how epigenetics or methylation controls which genes are turned on or off. Building on work following the Human Genome Project, we now understand that your DNA alone is not your destiny. Epigenetics can drive change in your cells. And importantly, it is believed that many of these epigenetic changes may be modifiable. Many researchers believe that emerging work in epigenetics may be critical for the development of more personalized medicines. An epigenetic clock is a biochemical test that is based on DNA methylation, which are chemical modifications of the DNA molecule. We now can reliably measure human age using a simple blood draw. By applying epigenetic clocks to DNA collected before and after a drug treatment, we're able to quickly determine if a drug is affecting the epigenetic aging process. Using these epigenetic clocks, we and others have found interventions that greatly reverse the age of mice and rats. Some of these results are expected to matter for human health as well. In 2019, Greg Fahey and I published results from a phase 1 human clinical trial that demonstrated a notable first, that a treatment consisting of already approved drugs and supplements could reverse all established epigenetic clocks in healthy older men aged between 50 and 65. In a current phase 2 trial known as TRIM-X, which is going on in California right now, we will assess if the same treatment can be applied to women and men between 40 and 80. The trial may also determine if the treatment leads to functional improvements in older individuals, such as increased leg strength that will delay onset of frailty. This ongoing work has provided a helpful template for the longevity research community. When used along with standard clinical and physiological testing, epigenetic clocks could add a rigorous and practical approach for determining if a new longevity drug is effective for use in healthy older individuals. Preventative medicine trials that previously took many years may now be completed in only 1 to 2 years, although tracking of longer-term health outcomes will be critical as well. The biotech industry is also now developing exciting new drugs targeting the biology of aging. As the data mature, there will be a need for a clear regulatory framework for drug approval in healthy older individuals. In addition, looking at current disease classifications to ensure they are inclusive of these new therapies would be very helpful. My hope is that this committee and others in government will recognize the recent biomedical breakthroughs, including biomarkers of aging, and modernize the approval process for new longevity treatments. We have an opportunity and arguably obligation to leverage these recent biomedical breakthroughs to identify interventions that may delay the onset of chronic diseases and which may revolutionize the field of preventative medicine. When we look back at past centuries, we find the high rate of child mortality completely unacceptable. Nearly 50% of babies born in the US in 1800 did not live past their 5th birthday. I predict that future generations will look back at our times and recoil in horror at the high mortality rate in the elderly. Globally, over 100,000 people die each day due to age-related diseases. We don't have to accept this anymore. Thank you for your time and invitation to speak to you.
Thank you. And at this point, we will now begin our first round of questions, and the chair will recognize himself for 5 minutes. Dr. Niedernhofer, there seem to be at least 3 active subfields of research here. There's cellular senescence, cell reprogramming, and thymic rejuvenation. There are probably others. Could you please briefly explain a bit about the status and goals of each of these and other approaches? Beginning with senescent cells, since you're one of the experts on that. And why don't you shoot for 3 minutes?
Well, thank you very much for this question. What the field has done has recognized what we call hallmarks of aging. So these are sort of the universal processes that seem to happen in every organism, from worm models to rodents, primates, humans. And these are all candidate targets for therapeutics. Senescent cells has emerged as one of those targets or hallmarks of aging that is very, very tractable. So this allows us to develop therapeutic drugs that target senescent cells. The reason this is important is senescent cells are cells that are stressed. They've responded to that stress in our body, which is a very healthy thing to do. It suppresses cancer, but afterwards, those cells linger chronically, and they secrete things into their environment, which are very pro-inflammatory. And it's quite clear that senescent cells at this point promote aging, as well as contribute to virtually every age-related disease. And so if we can clear them with drugs, help the body and the immune system clear them, we really see a tremendous impact of this approach. Reprogramming is a little bit different, where you're trying to rejuvenate through epigenetic changes, as Dr. Horvath has spoken about, to rejuvenate the cell rather than get rid of damaged cells. So this holds a lot of promise as well. There's lots of other approaches: rejuvenating stem cells, improving the response to genome instability, nutritional stress, and the response to nutrients. So we have lots of different opportunities here to target biology of aging, and I think they're all coming together quite nicely. A very important point to make is all of these hallmarks of aging are truly interrelated, meaning if we target any one of them, we can actually improve multiple of these hallmarks of aging and literally remove damaged cells or rejuvenate existing ones. And so there's a lot of promise here. Thank you.
Thank you. And I was struck by a study that you co-authored where healthy adult mice were dosed with extra senescent cells. And so can you just describe quickly what was the result of the senescent cells?
Yeah, that's a great question. So what's really important to prove cause and effect, which has been extremely challenging in the field of biology. We have these hallmarks of aging, but do they really cause aging? So what you have to do is add senescent cells if you want to test the hypothesis that they drive aging, or take them away. And so we have dosed animals with extra burdens of senescent cells, and the impact on those animals are they have reduced spontaneous activity, reduced grip strength, altered metabolism. So all of the things that we observe with normal aging can be driven by the transplantation of senescent cells into a model organism. And in fact, one of our studies showed that senescent immune cells are one of the most potent ways to drive aging of all of the tissues in your body. So we also have very strong pharmacologic as well as genetic evidence that if you remove senescent cells, you can regain the spontaneous activity, the grip strength, the endurance. So all of these things that we really are passionate about and lose as we become more frail with old age.
Yeah, thank you. And I find the senescent cells fascinating. Just the concept that they're these cells, they're not dead yet, but they're not doing their job. Until recently, it actually reminded me very much of the United States Senate. But it's not the subject of this hearing. Anyway, well, let's say, I guess I am close enough to the end of my time, and I'll turn it over to the ranking member.
I'll second the sentiment about the Senate. Well, thank you very much to our witnesses. It's been a really interesting hearing. Dr. Olshansky, I'd like to start with you. I was very interested in both your oral and written testimony where you talked about the potential cost savings of $38 trillion a year per year of extended lifespan. Could you talk a little bit about, in that analysis, how you came up with that number?
Actually, I didn't come up with that number. That was research that was done by some of our colleagues in the UK, some scientists at Harvard. That's a much longer story that I'm not the expert on having come up with the $38 trillion estimate. So I'm actually going to leave that question to the scientists that generated that research, but I'd be happy to send the citation or provide that citation to anyone who wants it.
Okay, well, let's talk about the potential other side of that equation, which, you talked about how currently the dealing with aging is like playing whack-a-mole, and that perhaps one of the, in your written testimony, you said perhaps one of the breakthroughs in geroscience would be disease compression, where we're compressing the period of our lives that we're experiencing these. But what if it's somewhere in the middle where we just get really good at whacking the moles, right, as we move along? But in that case, you would actually achieve greater human lifespan, but at much greater cost. And actually, I mean, that's kind of what we've been experiencing so far with our healthcare system, right? And this is, as a society, we've been really grappling with this issue of how to equitably distribute those costs, because it's great that we have these greater lifespans, but the treatments are becoming increasingly more expensive. So, why is, let me ask you to defend why you think it's a cost savings and not an increased cost, given the fact that either one would be preferable to what we've got now.
Yeah, so several years ago in 2018, I published an article in the Journal of the American Medical Association entitled From Lifespan to Health Span. And in there, I describe what we've done to ourselves. We've basically, in the 20th century, redistributed death from the young to the old. We've extended survival very effectively past the ages of 65 and 85. But we've done it against the backdrop of what of frailty and disability that rises exponentially with chronological age. I refer to that as the red zone, a time period where if you live into this red zone, frailty and disability is extremely high. If we continue along the pathway of attacking one disease at a time, in all likelihood, we will succeed in extending survival deeper and deeper into the red zone, which would raise costs per capita, would raise overall costs associated with healthcare. There would be a pretty heavy price to pay if we continue along the current path. The focus of geroscience is not on the line that pushes us towards later ages. It's on the red zone itself. It's to compress the red zone, push it to later ages so that when bad things happen that are associated with aging, they happen over a much shorter time period, and all the costs associated with that, with end-of-life care and end-of-life health issues, would be less costly and compressed over a shorter time period. And that is the kind of thing that we're seeing in the animal models, is compression of morbidity, not just lifespan extension.
Thank you. Dr. Niedernhofer, I had a question for you. I thought it was really interesting when you were talking about the societal implications of a greater lifespan. And you were careful to say this isn't your field of expertise, but I'm wondering your thoughts on some of the negative consequences. Because if you take the limiting case where human lifespan is greatly extended, then you get into a situation where every single birth adds to the population of the planet. So, I mean, what are the ethical implications of that? Because that would seem to be something that also society would struggle to deal with.
It's a great question, but I would really like to emphasize that our goal is to make the elderly more healthy, making them more useful so they're able to work, contribute, spend their money, vacation, do all those things that we really enjoy. So I don't see a negative consequence of this. Keeping people healthy active, more youthful, I think is all good for the economy, but I am not an expert.
Sure. Well, I mean, it's obviously this is something that we should do and need to do, but I mean, we also need to be realistic about the societal implications of it. All right. Well, thank you very much. I see my time's expired. I yield back, Mr. Chairman.
Thank you, and we'll now recognize Representative Perlmutter for 5 minutes.
Thanks, Mr. Chair. And this, maybe as I'm getting older, this is a very fascinating hearing we're having today. And we just had to move my mom into memory care. So, Dr. Horvath, I want to start with you. In terms of your biomarkers and your ability, the ability that's being developed to look at things across the board, so cardiovascular or cancer or brain diseases. So as somebody develops Alzheimer's, I mean, how are you seeing things that might help us reverse something like that? Because going back to Mr. Obernolte questions, we see sort of on a macro basis just huge cost to society, particularly from Alzheimer's. So can you sort of respond to that for me?
Yes, when it comes to Alzheimer's disease, I do see exciting results, certainly in basic research. I'm just at a conference here and I see wonderful, very promising talks. We really understand a lot about Alzheimer's disease. As our knowledge increases, we will undoubtedly develop effective medicines. I'm just as sure as you can be. Question is how fast. There is always a chance of serendipity, but I'm very happy to hear that there are ongoing clinical trials. What I can tell you, Alzheimer's disease, of course, is due to protein aggregation, so-called proteostasis, but also many other facets of aging touch it. The immune system, also epigenetic changes, changes in garbage removal or autophagy in the cells. And so, and there's really an army of researchers working on it, on all of these facets.
So,
and the same, of course, pertains to other diseases. Yeah. Mm-hmm.
So the other panelist is saying, look, if we are able to, through geroscience, start affecting how cells age, I assume that this would, as a general principle, help slow down the potential for an Alzheimer's or that kind of disease. I mean, are you seeing that in your research?
Yes. The idea is make cells more resilient. Young cells are resilient. They can tolerate various forms of stress. And so, yes, I think that's a very promising avenue.
Thank you. I'd like to, our problem as legislators is to figure out what are going to be the macro results of this. What are the consequences? And Dr. Niedernhofer, you started to opine on that a little bit. I mean, I think the good news is if we're successful here. I think you're probably right that we would have independent, productive, happy people. And that's my goal as a legislator, to improve society. So kind of give us what you think might be some of the problems that we as legislators would face if you as doctors and scientists are successful.
Problems that we would face, I think it would help solve a lot of problems. I mean, one example I'd like to give to you is think about centenarians. So it's a fact that they experience 20 plus years of healthy living. And in the last 2.5 years of their life, they use the healthcare system much less. They're not sick, they're not going to doctors, they're not taking as many medicines. And so biology tells us we can be fit, healthy, less frail in old age. Picture the Queen Elizabeth. I think she was a beautiful example. She's working, she's on vacation, and then she just has this very compressed period of morbidity. So I look at this as an opportunity to solve problems.
Thank you. I'll yield back to the chair. I think we'll get to do a second round, so I just need to digest all of this that you people are talking about. I would say one thing. We had a governor in Colorado, Dick Lamb, who was called Governor Gloom, some of you may remember, because he said everybody had a duty to die, and so that we didn't put extra pressure and costly pressure on the healthcare system. And you're saying that if this is successful, we're going to do just the opposite. So I appreciate that. I yield back to the chair.
Thank you, and we'll now, let's see, is Representative Byer still on the call? He was due up next, but if he is apparently not here, all right, 3-2-1, in that case, I think we'll start our second round. And so, well, let's say I was caught a little bit unawares here. What are the major federal players in funding anti-aging research right now? You know, I'm aware that the national academies have been ongoing for a while. The National Science Foundation has an effort going on, and so what are the major players, both nationally and internationally, in this? Dr. Niedernhofer, you want to take a swing at that?
Sure. So definitely the National Institutes of Health, in particular the National Institutes on Aging. I would also give credit to NCI, the Cancer Institute. They are thinking deeply about the interface of cancer and aging, as well as other institutes within NIH. The National Academy of Medicine has been very forthcoming in tackling aging. They've just written an incredible geroscience white paper that's available publicly. We see a lot of philanthropies trying to get involved. And I should also point out that the Office of the Director at NIH is investing in a lot of related projects analogous to the human genome, but more focused on aging. So for instance, the SENNet or consortium that's addressing senescent cells, trying to characterize them so we can ultimately develop better therapeutics targeting them. So I think there's a lot of opportunity. I think what's really important for legislators is to keep this democratized. So this is not something that's just swept away by wealthy individuals, but really we learn about the biology of aging across very diverse groups of individuals so that everybody can benefit from this research.
Now, are the majority of the candidate drugs involved as senolytics? Are these small molecule off-patent drugs that are likely to be fairly cheap to provide, or are they're going to be any feeling on how expensive these would be?
That is a great question. So right now we have largely in the field, particularly academics, taking financial support, federal funding for our research. We've really focused on natural products as well as repurposing existing drugs. And the main rationale for that is these are going to be faster to test in a clinical setting because we can jump right in with a phase 2 clinical trial and not have to start from ground zero where we're just proving safety. So this is speaking to the urgency that we recognize. They are relatively inexpensive because these are either drugs that are FDA approved but not necessarily used or off patent, or natural products that are relatively inexpensive. So the one I can provide actual numbers on is we've been working with the Seton. It's advanced to clinical trials. It's about $15 a dose, and you need 2 doses every 2 weeks. So quite inexpensive.
Yeah, because one of the things that we wrestle with policy-wise is that for a drug which is off patent, there's very little commercial incentive to go and pay for the clinical trials. I was just wondering if that's something where we're actually just going to need government money to make sure the promising drugs are carried through to clinical trials because of the absence of a commercial incentive.
I do think it would be the responsibility of the federal government to get this started. I think it's also really important to de-risk this approach. As Dr. Horvath was saying, it's a challenge right now because of the regulatory system as well. We don't have a way to approve a drug to extend healthy aging. And so proof of principle and advancing some of these inexpensive therapeutics that everyone can access. It would be fabulous to have federal support for that.
Thank you. And so I guess I will just yield to the ranking member at this point.
Thank you, Mr. Chairman. Dr. Horvath, I wanted to ask about a part of your testimony. You were discussing that there will be a need for a new regulatory framework for the treatment of healthy people, as opposed to our current regulatory framework that's geared more towards treating people who are ill. I wondered if you could, that's very much our bailiwick, and it's something that's going to be useful for us to start thinking about. Can you talk about what a framework like that would look like and how it would differ from the existing framework that we have?
I think it would be important to develop different metrics of measuring success. And the reason is, imagine you have a drug that you give to a middle-aged person, a 40-year-old, with the hope that you will prevent many future diseases. But then you, of course, would have to follow this person for decades, and it's cost prohibitive. So in order to advance medications that prevent the onset of these age-related diseases, we need to find surrogate metrics as opposed to actual disease states. In other words, biomarkers. And it would be very good if a regulatory agency could take the lead to organize a panel and really carefully look at the data, generate additional data, and develop surrogate markers, what we call surrogate endpoints, that are trustworthy, that most scientists believe in. But then also, these should be available then to the biotech industry so that it unleashes investment from private sources to fund these trials.
Right. Actually, Dr. Niedernhofer, let me ask you about that as well, because in your testimony, you were talking about one of the barriers to the advancement of geroscience being the lack of aging biomarkers. And it seemed like if we had, I think that's exactly what Dr. Horvath is discussing, you know, that we would need these aging biomarkers to be able to prove the therapeutic benefit that would need to fit into the regulatory framework of healthcare. So could you talk about what those biomarkers might look like and how we can eliminate that barrier?
Yes, and I have deep respect for the biomarkers, the epigenetic biomarkers that Dr. Horvath has developed. I think they are some of the key tools in our toolbox, but we need more. And I think it even stems from trying to assess what people value in old age across very different ethnic and socioeconomic groups. Just understanding what they value, what their goals are, starting there. So there could be functional tests that we have, how quickly you walk, how quickly you get up and down out of a chair, what's the slow decay in your ability to do a lap around the track. It should also incorporate molecular markers that are very quantitative, and we have a lot to learn here, but I think it should start really with a conversation between regulators, what they need to prove safety, efficacy, and then the scientists who can actually figure out how to measure those and develop those tools. So I agree with Dr. Horvath that it'd be lovely to have panel discussions where we really anticipate what's going to be needed to compress the length, the duration, and the size of these clinical trials so that we can afford them, and do it in an iterative process, maybe even in parallel, so that we're able to test many different approaches and really come up with an answer to extend health span as quickly as we can.
But just playing devil's advocate, though, I mean, I think we're talking about something more than just measuring a deterioration that occurs with aging. And I mean, for example, well, I'm feeling very old this morning because I had congressional football practice, and my body's reminding me that it's not the same body that it was 30 years ago when I actually played football. But I mean, you could measure my time running around a track, and it certainly has decayed in the last few years. But the whole purpose of a therapeutic treatment is so that that will not decay, right? So you wouldn't have that marker right? So we need something cellular that provides, a marker for the efficacy of the treatments that we're proposing to be able to fit into the regulatory framework we have. I mean, don't we?
Yeah.
We absolutely do, but I do believe as well that you need to tie it to some functional outcomes so that you really, the individual is experiencing the benefit as well.
If I could add a few comments. Aging really starts very early on. On a molecular level, children already age in some shape or form. And so it is correct, we do need these early molecular biomarkers as well. I want to add another point, which is when you put two scientists in the room, you will get two different opinions. They will never agree on which biomarkers should be used. So there's really a need for leadership, regulatory leadership, impartial leaders making decisions, executive decisions, informed decisions, decisions based on data, rigorous testing, but leadership would help the field tremendously.
Right. Well, thank you very much. I yield back, Mr. Chairman.
Thank you, and we'll turn it over to Representative Perlmutter.
Dr. Olshansky, I've got a couple questions for you. So this morning I was going through my news feeds, and one of the articles that pops up is 90 will be the new 40 in 10 years. Which again, as I'm, at 69, sort of marching, time keeps marching on, that was music to my ears in looking at that. But there are, do you think that's a legitimate headline? And to the other panelists, you can answer that too, but my second question would be more, again, going back to the macroeconomics of this, to Social Security and things like that. If in fact we are moving forward where people can live longer, healthier, more productive lives, then there are some things on a macroeconomic level that we need to prepare for, one of those being Social Security. So I'll just let you respond generally to both of those questions.
Yeah, so I'll respond to the second one first. So look, when geroprotectors come online, they will indeed successfully produce more healthy older people. There will be more people surviving past the age of 65, 85, 90 than any time in history, in part as a result of these therapeutics. So Social Security will be challenged. There's no question that the Social Security Administration is going to have to deal with a larger population that will be drawing benefits for a longer time period. However, if we're extending healthy life and people decide to work longer and it will be their option, you will now have justification for delaying age at entitlement. Whether we're pushing back 62 and 67 by how much, there would certainly be logical justification for altering age at retirement, and that would certainly deal with the issue associated with more healthy older individuals drawing from Social Security. I saw the same story you saw, by the way, on 90 is the new 40 in 10 years. It's the 10 years, by the way, that bothers me. The 10-year claim has been made for 2,000 years, that some magical breakthrough is going to happen in exactly 10 years. The 90-40 might be a bit of an exaggeration, but the concept, I think, is right on target. And that is, it will take a longer time period to grow old biologically. Maybe, it'll take 90 years to become 70 or 80 years to become 60 or something along those lines. But practically at our level, the way in which you and I and all the listeners operate, we will experience biological time at a slower rate. A year from now, you will not be a year older. You might be 9 months older or 8 months older. It's not going to stop aging. It's going to slow it down. And in the end, that's what we want, is to retain our youthful vigor for a longer time period. So some of the numbers, I don't like this embellishment and exaggeration that I see in the field all the time, but the overall premise, I think, may be on target.
Well, I'm smiling because we have this thing at the YMCA called the E-Gym, where it's sort of traction, all this stuff, and it has biological age. This makes me very happy when I look at it. It says my legs are 36 years old, my core is 35, and my chest and arms are 27. So to any future employers, I give them this, say, look, there's a lot of years left in this guy. So, but I think what you're all talking about is that there are potential scientific breakthroughs, whether it's through the use of biomarkers to kind of manage things, or metamorphin, or whatever the drug was that was mentioned. But there are also other things that we're becoming, smarter about in terms of nutrition and exercise and those kinds of things, which, again, Dr. Niedernhofer, we're trying to both have quantitative, objective kinds of markers, but also there's a qualitative element to this. Does it hurt every time you get up out of a chair? So I don't know if you have any comments either to my biological age, which I think is pretty good, or otherwise.
Well, I would say congratulations, and we need to continue to build these tools so that we can do this for everyone and accurately. I don't completely trust the tools that we have entirely at this point.
Well, I do.
Obviously, you're good. You're on a great trajectory. The other thing I would just comment about, sort of in terms of the economy, is just picture a person who's in memory care versus a person who's independent, which I think is what geroscience approaches can achieve, a little, an extended period of independence and activity. So they are going to contribute one way or another to the economy, much more so than somebody who's sadly trapped in memory care.
Thank you. I yield back to the chair.
Thank you. And I believe we can actually have another quick round of questions because this remains fascinating. And I will now recognize myself for 5 minutes. What is the current understanding about the evolutionary advantages for senescence? Because that has me really confused. Apparently some species like lobsters do not have senescence and then others do. What is believed to be the advantage of why this has evolved?
I believe this is a question for me.
Go for it.
Thank you. So senescence evolved as a tumor suppressor mechanism. In my mind, it is one of the most anti-cancer programs we have in our body. So it's necessary for multicellular organisms that live a very long time. So if a cell is stressed, in particular the DNA of that cell is stressed, that cell will respond by activating signals that say, I will never copy myself again and make a new cell. And that prevents that damaged stress cell from turning into a tumor. So it's very advantageous. We also know that there's senescent cells in a number of physiologic healthy contexts. So during wound healing, obviously there's a stress to your skin if you're cut open, and senescent cells will accumulate at that site to help heal it. But these are very acute events that are cleaned up and help you carry on without having the chronic inflammation that can come with building up senescent cells with aging. We think part of the problem with senescence in old age is the cells just accumulate because your immune system, the function of it declines a bit with aging, and therefore you're unable to receive signals from the senescent cells that call in your immune system to clear them. So it's a lovely cancer protection mechanism, contributes a lot to various physiologic states in mammals, but turns against us as we get older, as many things do in biology of aging.
So do you see the mechanism throttled down in elephants versus mice, or is there a pretty universal pattern there?
So that's a great question. So we've learned a lot from comparing different species, and indeed elephants are some of the longer lived species. They have extra copies of genes that reduce their cancer risk, and therefore they just experience much less cellular senescence. But there's still a lot to learn in this space. And I think we're a little bit caught up in just lack of definition of a true senescent cell. It's very hard in my mind to talk about healthy senescent cells and pathologic senescent cells and not get everybody confused.
Yeah. Was there someone else who wanted to comment?
Yes, yeah, if that's okay. So really good questions, by the way. So this issue of humans and elephants and dogs and how long we live and why it's all relevant is actually central to the study of aging and longevity. But keep in mind, we cannot have aging or death programs that evolved within us. We don't have a ticking time bomb that goes off at a certain time period. Natural selection could not have led to the evolution of ticking time bombs in our bodies. So think of aging and senescence as an inadvertent byproduct of fixed genetic programs that exist for growth, development, and reproduction. And that explains why different species live different lifespans. You know, dogs live, about 15 years, and they go through puberty at 9 months. They go, their reproductive window is much shorter. Our reproductive window is longer. We live longer as a result. So there's this calibration between duration of life and the reproductive window of the species. But importantly, there isn't a death program. And the absence of a death program is the reason why we can intervene. It's the reason why these geroscience interventions are going to work. And it's also the reason why diet and exercise can actually have an influence on how healthy we are and how long we live as well. The field is wide open for intervention.
Now, are there examples of where the extension of the health span is not associated with the extension of lifespan in any animal studies or so on? Are these, it's a very, politically it solves a lot of problems if the main result of all these treatments is that you're healthier longer and then you die at pretty much the same time. If that's not really the case, then it's a much more complicated set of policy implications. So what's the best understanding of that relationship?
I can comment real quickly. I think that when we introduce these geroprotectors, we know that we will see a compression of morbidity and disability. By how much exactly, I think we cannot yet determine. We will need the biomarkers to make that determination. The unanswered question, which I think is the one that you're addressing, is how much longer might we live as a result? And we don't know yet how much longer we will live as a result. And keep in mind, when scientists and others come along and suggest we're going to live 10, 20, 30, 40, 50 years longer as a result, there's no evidence to support that at all. We cannot, that is an untestable hypothesis on any sort of radical life extension proposal that folks are making. So that's one of the reasons why we're focusing in on health span. It's something we can measure and we can detect very quickly in these types of studies.
So I guess I was asking about animals. Any hints from smaller animals? Dr. Horvath, I guess.
Yeah, I've looked very carefully at epigenetic determinants of what I call maximum lifespan in species. We have analyzed 348 mammalian species, from maximum lifespan 2 years in a shrew to the bowhead whale, whose maximum lifespan is 211. And there is a strong epigenetic signal. But the interesting finding was that whatever determines maximum lifespan was actually quite different from what relates to human mortality risk. So my opinion is that the determinants of maximum lifespan are quite different from what we care about here, which is health span.
All right, thank you, and I'll yield to the ranking member.
Thank you, Mr. Chairman. Dr. Niedernhofer, we were talking a few minutes ago about the barriers to the advancement of geroscience that you'd brought up in your testimony. And one of the barriers that you mentioned is lack of public knowledge. Can you talk a little bit about why that's a barrier and what we can be doing about that?
Yes, thank you for asking. I just feel that it's a little bit of a rarefied crowd that really understands geroscience at this point. And I feel like we need to gather a lot more information from various stakeholders in the United States to understand what they value in old age and how we can address their needs as well. This may not be for everyone. I think deeply about Native Americans who can't wait to get old and be an elder in their community, and they don't want to interfere with that process. So I think we need to gather information, and educate people to get buy-in, because I think there's an awful lot of people who would be tremendously excited about it, but we need to understand across our population who, what the various opinions are about this approach.
Mhm. Well, I mean, I think also maybe caution is warranted because, as Dr. Olshansky just pointed out, we don't have any scientific evidence that we can extend lifespan. We just know that we have these cellular epigenetic clocks that we think we can eliminate. And we also think that maybe we can reverse some of the effects of aging, such as Alzheimer's disease, which would have by itself would have a huge benefit for society. But we really can't promise people anything. And I actually think that when people hear about this, they could be tremendously excited. It's just that we have to be very cautious about being realistic with them about what we can and can't promise. So anyway, I thought that was interesting. But that would, I mean, to your point, that would help eliminate some of the other barriers that you brought up. Certainly lack of scientists. If we evangelized, this emerging field better, we could get more young people in training in fields to be able to do this kind of research. Lack of funding, certainly, a greater public awareness would help us with that. Dr. Horvath, the chairman was having what I thought was a really interesting discussion about clinical trials and how they're funded, given that many of the drugs that we are investigating for the use of geroscience are off patent, and that eliminates a profit incentive for private industry to fund those. So, we're funding research through NIH, we're funding research through NSF. Do you think that this is a barrier to actually bringing therapeutics to market? And if it is, then how much more funding do you think we would have to do?
Yes, just to echo what you said, it would be wonderful if we found incentives to repurpose existing drugs or drugs that are off patent, natural products. Now, I'm thinking about how to do it, I really don't have a good solution, unfortunately, but yes, so many scientists really work on that. I want to say most scientists in the geroscience field actually work on that. Why? Because natural products are safe. Also to remind everyone, when you go back to a dinner party and talk about aging, everybody will share their favorite supplements with you. What are they taking? Vitamin D, whatever. And what is so frustrating to me personally is everybody has their favorite one, but the evidence is so weak. And it would be wonderful then if we found ways to do rigorous studies of these supplements that so many of us are taking. And yeah, I'm not sure whether I answered your question.
Well, it might be a problem that solves itself, because once we can state credibly that there are health benefits to doing this, there's going to be incredible public interest and public demand for this. And so that's going to create a market void for companies to fill. And that'll be a source of private funding. So we'll see. Well, it's been a fascinating discussion. In closing, though, I want to comment on Congressman Perlmutter's biological age, and just point out how much we've enjoyed having him here on the committee. And since the biological age is so low, you definitely should not be, sir, retiring from Congress. I yield back, Mr. Chairman.
That's right. Well, yeah, we'll be using the Yamanaka factors to clone Perlmutter. So it's all underway. Okay, Representative Perlmutter.
All right, I thank the gentleman. You have used, all of you, a euphemism of compression of morbidity, which I'm not, as a lawyer, I'm going, okay, what the heck does that mean? Does that mean when you die, it happens quick? Or, I mean, Dr. Olshansky, I think you've used it the most. So tell me what it is you mean by compression of morbidity and why that's an important concept.
Yeah, great question. So it basically means everything that goes wrong with our bodies and our mind with the passage of time would be delayed. If normally you would see an expression of a particular disease or disorder at age 60 or 70 or 80, it might be delayed to 70, 80, or 90. And when things go wrong, they would happen more rapidly. And so actually it's a fairly straightforward concept, and it's not foreign to us because we already see it among subgroups of the population that exists today. It's about 15% of the US population called superagers. And these are individuals that make it out past the age of 80, cognitively intact. You can't really tell the difference between them and a 40 or a 50-year-old. And so we see it today among subgroups of the population that are already experiencing morbidity and disability compression. They're healthy, they're active, they're exercising, they can be president, they can be CEO, they can do whatever they want at 60, 70, 80, 90, and it's already here today among subgroups. So it's what we want. It's a healthier life for a longer time period, and basically age becomes just a number. It becomes largely irrelevant. And when things go wrong, they go wrong quickly. And so it's not going to stop us from aging. It's not going to stop us from dying at some point, but it will lead to a longer period of youthful vigor. And in the end, certainly that's what my father told me when he was in his 90s. He said, all he wants is his health. And I couldn't agree with him more. It is the most precious commodity. And that's what this is all about, is extending the amount of this precious commodity that all of us have in life.
Well, I want to thank you. I want to thank your fellow panelists. This has been fascinating. I think that's a good place to close. I'll yield back to the chair.
Thank you. And now, before we bring this hearing to a close, I want to just thank our witnesses again for testifying before the committee. The record will remain open for 2 weeks for additional statements from the members and for any additional questions the committee may ask of the witnesses. And the witnesses are now excused, and the hearing is now adjourned.